evening, all of you. बैठ जा. कौन लगाएगा? लगाना यहाँ पे. क्या नहीं? गजू लोग कहाँ गया? दुर्वेश कहाँ गया? लगाओ. But it's broken, huh? Careful. चलो. What did we study the other day? We are already finished with experiments on osmosis. Why the rest? What happened to the other? Huh?
Inter-school competition. Inter-school competition.
In your school? Not going? They have gone from your school, no?
हमारे सीनियर से
Last bench, can you see? Last bench, can you see?
Yes.
So today we are going to start with, we are studying absorption values. Some concepts that we are going to study today would be, first of all, what did we study last? We had studied three concepts the other day. We had studied about what? Imbibition, we had studied about diffusion, we had studied about osmosis, various different experimental concepts about osmosis. What was the definition of osmosis? The last one. पूरा किचन उठा के लाया है वो. क्या गिरा तुम्हारे बैग से? कितने लीटर्स वाटर थे इसके अंदर? What is the definition of osmosis? Jhandeep. जो भी है वो दीप का अभी तक जल नहीं रहा है ना, इसलिए प्रॉब्लम यही है. उसके पेरेंट्स ने दीप रखा है नाम ये सोच के कि बहुत अच्छी तरह से जलेगा, ये तो जलने का नाम ही नहीं ले रहा है. वो दीप हटा दे तेरे नाम से. चलो बोलो, what is the definition of osmosis? I cannot shout also because he is teaching it. बोलो. सोच रहा है. What is the definition of osmosis? I'm not asking you. I don't want to know from you. जब मैं पूछूंगी, you answer. बोलो. I'm giving you one more chance. What is the definition of osmosis? पेपर में कुछ तो लिखोगे कि कुछ नहीं लिखने वाले. What is the definition of osmosis? Very good. When we are talking about osmosis, remember the basic difference with osmosis and diffusion is you are to very much focus on the word, the movement of the water molecules or the movement of the solvents molecules. And the rest almost remains the same. We are talking about the movement from a region of high concentration to a region of low concentration. If you are talking about osmosis, then you have to mention that it is through a barrier, and the barrier is known as selectively permeable membrane, semi-permeable membrane. And if you are talking about diffusion, then what should you write, Prisha?
The movement of from high concentration to low
When the molecules are in direct contact. When the molecules are in direct contact. So ये है basic differences, याद रखना है. We had learned many examples, many experimental concepts about osmosis. Now, one more thing we are going to study here is that based on the concept of osmosis, the other day I had just introduced this topic. Say suppose I talk about a solution. I talk about a solution of unknown strength. I am talking about a solution of unknown strength. Unknown strength, what do I mean by that? Unknown strength means I don't know whether the solution is 20%, 30%, 5%, 10% strength. I don't know anything. But yes, we are experimenting with certain type of plant cells or animal cells. Any of these we can take. So right now, I'm talking about plant cells. Right now, I'm talking about plant cells. So I take some plant cells, and these plant cells, they should be having a vacuole. I'm talking about the plant cells, and the plant cells should be having a vacuole. The plant cells should be having a vacuole. So now that I talk about the plant cells that we have put and the vacuole inside, try to understand step by step what is happening here. First of all, when I'm talking about this, in this type of solution, I said this is a solution, but we don't know what the strength of the solution is. But yes, it is just sugar solution, salt solution, only that much we have to understand. And what is the setup? The rest of the setup is same solution, same unknown solution given in 3 different beakers, and you are inserting some plant or animal cells. You observe for a particular period of time, and after a particular period of time, you are to study, you are to conclude with the observation. So after a particular period of time, in the first beaker A, what's happening? The cells that we have put, the cell shape, what about the cell shape? The cell shape remains the same. What about the second? In the second, perhaps we get to see that the cells have bloated up little bit. The cells have bloated up little bit. Now I draw the diagram something this way, but remember the bloating of the cells again is not very remarkable. Little bit. If I have to talk about a period of some 8 to 10 hours, little bit. So here we are talking about then what about in B? What about the cell shape? The cell shape is wallet. C, what about the cell shape in the unknown solution? In C, we get to see that the cells have little bit shrunken. The cell shape is little bit shrunken. So first is this, which you can observe. Now we have to understand why should this happen? Why should this happen? In one, the cell shape remains the same. In the other, swollen, and in the other, shrunken. The reason must be something like this, that if I'm talking about that the cell shape remains the same after a particular period of time, it must be something like this, that certain amount of water must have entered, or whatever amount of water must have entered, whatever amount of solvent must have entered, and whatever amount of solvent must have moved out is perhaps the same. That's the reason after a particular period of time, the cell does not show any remarkable change in its shape. Then when is this possible? When is this possible that I say that if 3 molecules of water is entering, maybe just 1, 2 molecules of water must have moved out. Or if I'm saying that 6 molecules entering, maybe 4 molecules moving out. So I should say that the net movement of water molecule, the net movement of the water molecule into the cell is something negligible, 0 it is. The net movement is 0 because whatever is entering is almost the same what is exiting out. What about this side? Why should the cells swell up? Now that's very remarkable. Here we are saying, why should the cells swell up? The cells must be swelling up because the number of molecules entering is much more than what must have come out. So we are taking this as remarkable process, that the number of water molecules are entering in large amounts. So many water molecules are entering inside the cell, and that's the reason after a particular period of time, the cell swells up. So here what is happening? The water, what type of movement does the water show? Water movement. Okay, I would write. What about the water movement here? The water movement is inward movement, and inward movement, what will we say? This is also known as endoosmosis. Inward movement is also known as endoosmosis. We'll learn about the properties of the solution a little bit later. In the third, I said, so far the shape what? It is shrunken. Then what should I say? What about the water movement? In case of the water movement, I should understand the movement is outward. Outward movement of water. Outward movement of water. And the outward movement of water should also be written as, or should also be understood as, exoosmosis. So here we are learning two words, endoosmosis, exoosmosis. The other day we had already studied about osmosis. Now we try to understand that what are the two types and under what situation does it happen. What are the two types? If the water enters inside the cell, endoosmosis. And this is what should happen for a cell. If the water exits from a cell, the cell will progressively become shrunken, and that process would be termed as exoosmosis. Two types of osmosis. Now here we try to understand that what should be the properties of the solution. Why in certain case the water is entering and why in certain case the water is leaving the cell, and why in certain cases there is no change absolutely. The first one, if you try to analyze, we may find that in the first situation, in the first condition, in the first setup, whatever amount of water is entering, almost the same like what is water coming out, that is perhaps because the strength of the solution and the strength of the sap are almost the same. The strength of the solution and the strength of the sap, meaning whatever amount of salt, water, sugar, water inside in the sap and the composition outside, or the strength outside, I'll not say composition, the strength outside is almost the same. Is almost the same. Then what type of a solution we are going to name this as? What is the word to be used? Such type of a solution is known as isotonic solution. The definitions you have to know now. In an isotonic solution, the net movement of the water molecule is zero. There is no remarkable loss, there is no remarkable gain by the cell. What is the definition of this word? Isotonic. In this case, whenever you are defining these words, I will ask you to write down the definition after some time. Please listen carefully. Isotonic solution. You are defining the solution with reference to the cell sap, remember. Relative definitions this has to be. Meaning you are writing one in comparison with the other. Relatively you are writing that. Let me define what is isotonic solution. The solution which has same amount of solute and water as compared to the cell sap. I'm talking about a plant cell now. No? So I'm saying the solution which has same amount of water and solute. This is the solute, no, now, yeah. Same amount of water and solute as compared to the cell sap. Let us go to the next. In the next, what is happening? Endoosmosis is happening. Why should the water constantly keep on entering? Because this medium is dilute and this medium is concentrated. So now what is the term used for the solution? The term used for the solution would be hypotonic solution. Hypotonic. Now how will we define this? What is a hypotonic solution? A solution which has higher concentration of water and lesser amount of or lesser concentration of solute as compared to the cell sap or as compared to the cell. Hypotonic. So this solution is hypotonic. And what about this sap?
In the textbook it is given for endoosmosis.
First, you be very sure what you are asking. Bolo. Should I proceed?
Yes.
Now, when we are talking about hypotonic, what is the meaning of hypotonic? Try to understand the concept. I am talking about a solution outside the cell, जिसमें water ज़्यादा है, solute कम है. How do I measure? यहाँ से एक वॉल्यूम लेंगे, यहाँ से भी एक वॉल्यूम लेंगे. This is called volume by volume analysis. So volume by volume जब analysis होता है, then it is understood. ये जो solution है, this has more water, less solute, and ये जो cell fat है, this has more solute, less water. So can we say that water is moving from a region of high concentration to low concentration? Obviously no. यहाँ पे मैंने अभी बोला है कि यहाँ पे water ज्यादा है. Now water is moving from a region of high concentration to low concentration. आ रहा है समझ में? So what is the definition of hypertonic solution? Now what should we write? A solution which has higher concentration of water and lower concentration of solute as compared to a cell. Cell sap या cell दोनों करेक्ट है, पर cell sap लिखोगे तो और अच्छा है. So हमेशा cell के साथ compare करके लिखना है definition. I said this type of definition. If I write the definition something like this, is my definition correct? Hypertonic solution is a solution which has higher concentration of water and lower concentration of solute. This definition is not complete. You will not get full mark for this definition. You may not get a mark also for this definition. क्योंकि this definition is not a complete definition. Tonicity जब भी हम पढ़ते हैं, tonicity always has to be related. दो मीडियम को हम कंपेयर कर रहे हैं, always related. ठीक है? So ये हो गया hypotonic. Let us talk about the third condition. So what is the type of osmosis happening? In a hypotonic solution, cells will show what type of osmosis? Endosmosis. Come to the third. यहाँ पे क्या हो गया? The cells after a particular period of time they have started showing little bit of shrinkage. तो इसका मतलब क्या हो रहा है? What must have happened? Shrinkage क्यों? Because the amount of water lost must have been too much as compared to whatever little bit of water they must have gained. As compared to what little bit of water they got, they have given too much. They have given away too much of water. That's the reason the process of exoosmosis. That's the reason the process of exoosmosis. So this is what we are talking about. If it is exoosmosis, the cell is progressively losing water. Then what type of a solution this should be? In what type of a solution is it in now? It is in a hypertonic solution. It is in a hypertonic solution. Tanmay, define the solution now. Where is your brother gone? Where is the other one? हाँ?
Modeling.
अरे
Lamp walk.
वो तन्मय
मैं king बना king गलती से.
अरे बाप रे
अभी he is representing our school.
Model है model.
what a disgrace for the school. चलो ना
That's why you lost. Maybe lost.
that's why you lost.
हम वो English के लिए कुछ और ही बोलना चाहते हैं.
Ma'am, we asked you questions on current affairs.
अरे
मैं वो बीच में पढ़ाई होना शुरू कर दिया.
करंट अफेयर्स को तो आसपास नहीं जाता होगा है ना. So now you understand that most of these models they actually don't have brains. So now when we talk अभी देखो ये सबसे इंटरेस्टिंग टॉपिक है. क्या दी है? शायद अभी मैं ऑस्मोसिस पढ़ा रही थी तो इधर उधर देख रहा था जैसे ये टॉपिक शुरू किया इतना इंटरेस्टिंग टॉपिक है ना. चलो now I ask Tanmay define this word hypertonic solution. Only one Tanmay अभी है क्लास में.
A solution in which the concentration of water is more inside the cell.
Inside the
Cell.
तो तुम सॉल्यूशन डिफाइन कर रहे हो inside the cell कैसे? So defining this solution ये सॉल्यूशन की डिफाइन करो. I want to understand hypertonic solution बताओ.
Solution is less than the solute.
Complete बोलो.
Solution is less
Hypertonic
than
solution is one in which the concentration of
solution is less than the concentration.
दो चीज तुमको बोलना है solute and solvent. आप फिर से बोलो.
Hypertonic solution is the solution in which the concentration of solvent is less than the concentration of solute compared to a cell size.
So we are talking about hypertonic solution जहाँ पे water कम है and solute ज्यादा है as compared to the cell. That is the reason why the cell is constantly releasing out water. Why is the cell releasing out water? Trying to bring about an equilibrium. Trying to bring about an equilibrium on either side of the semi-permeable membrane. अब अगर either side of the semi-permeable membrane, if equilibrium is reached, then the process of movement of water molecule will stop. But generally, experimental situation में ये लाना बहुत difficult है. So when we are talking about hypertonic solution, now remember the definitions: a solution which has higher concentration of solute, less concentration of water as compared to cell. Hypertonic solution which has higher concentration of water, less concentration of solute as compared to cell. Isotonic solution, a solution which has same concentration of water and solute as compared to a cell. Because all the three definitions one by one we will write down, then I move on to the next topic. Remember these definitions. You are always asked comparisons to write, definitions to write. Complete writing down the definition comparatively with reference to a cell हमेशा लिखना है. Always with reference to a cell you are supposed to write. Sometimes you are getting questions in your paper. Sometimes you get questions in your paper instead of this plant cells, they may also take what RBC cells that also may be given in your paper. Concept remains exactly the same. Concept will remain exactly the same. There will be no difference at all. It is something like this. When I talk about the three different conditions, see here, I may talk about a situation like this. Why should the cell shrink? Why should the cell remain the same? And why will the cell swell up? Either you talk about a plant cell or an animal cell, the properties are more or less the same. The cell's observation will be more or less the same. Write down the definitions. I'll give you 2 minutes time. Endosmosis, exosmosis, you have to remember the definitions. The inward movement of water, outward movement of water. Endosmosis and exosmosis. Endosmosis and exosmosis. No, you are writing on your own. Before, you should be able to write. 3 definitions: isotonic, hypotonic, hypertonic. Book देख के नहीं लिखना है. You are trying to write on your own. Done. देख के नहीं लिखना है. You understand and then you write. It should hardly take 2 minutes, not more than that. हो गया? Finished? No, all of you done? Now, next is we try to understand a similar setup that already we have come across, but little bit with a change. Now here we are talking about very same beaker and a solution in the beaker. I'm sorry, water in the beaker, and then the thistle funnel and a particular type of a solution in the thistle funnel. So it is a sugar solution or the salt solution in the thistle funnel. So I may also talk about a specific strength of the solution. A specific strength of the solution, 5% to the solution, 15% to the solution. But here the difference in this setup as compared to the rest is, now here we are also to add or insert a piston and apply a weight over the piston. Now, of course, we are to attach this with the clamp and the stand. We have to attach this with the clamp and the stand. So here, this piston is something same like those pitchkaris that we use during Holi, you know. Exactly same way. We are talking about the thistle funnel, which is acting like the entire funnel shaped tube. And then we have got a tube-like structure, and over that some weights placed so that there is an amount of pressure being exerted on the upper surface of the solution of the funnel. So this is the only difference as compared to the other experiments on osmosis that we have studied. What is the difference? Apart from the solution in the funnel and the water in the beaker, what are we also taking? We are inserting a piston in the funnel and exerting a weight on the solution. Now, because of this, we get to see that after a particular period of time, say suppose after about 8 to 10 hours, what is the observation? After about 8 to 10 hours, we get to see that the level of the solution in the funnel remains exactly the same. Level of the solution in the funnel remains exactly the same, and the level of the water in the beaker that also remains exactly the same. कोई भी change नहीं होता है. There's no change at all. But what do you think? What should be the directional movement of the water? The directional movement of the water should be same like this. Yes or no? Yes. The directional movement of the water should be same like this, because here we are talking about higher concentration of water in the beaker. And in the funnel, we are talking about a solution. So obviously, you know the rule of osmosis, which says water moving from a region of higher to a region of lower concentration. But यहाँ पे क्यों? There is no movement of the water at all. And after a particular period of time, we get to see that everything is as it is. Why? What do you think must have been the reason?
Because the pressure exerted by the water and by that reaction.
One at a time. Huh?
The pressure exerted by the water on the membrane and the weight on the membrane.
Okay.
The weight on the piston is equal to the osmotic pressure. So it
Nullifies it. What to understand from here? Of course, there is a number one. Of course, there is a concentration difference. And if you go by the concentration difference, then water should have entered into the funnel. Is it happening? No, it is not happening. Why is it not happening? It is not happening because there is a pressure which is exerted on the solution, and this pressure is also exerting on this membrane, that is the cellophane or the parchment, whatever you consider, on the cellophane or the parchment membrane. And this is what is not allowing the water movement this side. This is what is not allowing the water movement this side. There is a pressure exerting this way, which is not allowing movement this way. What is this pressure called? This pressure is called the osmotic pressure. What is this called? Osmotic pressure. And how do we define osmotic pressure? What is osmotic pressure? The minimum pressure which prevents the movement of the solvent into a solution. The minimum pressure which prevents. Now, are we saying that the solution is coming out from the funnel into the beaker? No. वो नहीं आ रहा है. क्यों? दुर्वेश क्यों नहीं आएगा? Why will the solution not come out? इस तरह का movement होगा क्या? Will this type of a movement ever happen? क्यों नहीं?
because the membrane is semi-permeable.
You are not doing it. He is doing it.
So,
Because
Category is semi-permeable.
it is semi-permeable. And
Selected particles are down.
it is semi-permeable, तो solute तो वैसे भी आएगा नहीं. And since there is a concentration difference, water भी आएगा. But now we are talking about that if there is a pressure, then also why the water not coming out? If there is a pressure, water is not coming out because it is a semi-permeable membrane. Had this been the muslin cloth or something, then yes, water would have come out because of the pressure. So here we are understanding that what is the concept of osmotic pressure. Whenever such type of a situation happens, even on either side of the semi-permeable membrane, if there is concentration difference, but at one side there is an application of pressure, the process of osmosis will be stopped. The process of osmosis will not take place. Why? What is the pressure responsible for not allowing osmosis to take place? Osmotic pressure. What is the definition? The definition is minimum pressure which prevents the movement of the solvent. This is solvent, no? The movement of the solvent into the solution. So that is the reason neither the level of the solution is increasing nor the level of the water drops. Try to complete these questions now. Osmotic pressure. Look into the questions.
Orally.
Look at the questions. I'm going to ask you after some time. See,
Orally.
orally. Look into the question. Get prepared with the answer. All the four. Read one by one. और that much is its flexibility. इसके बाद इसका joints टूट जाएगा. Come this way. I'll read out the question. Number one. The setup is the same like I showed you here. Only difference is they are writing sucrose. Sucrose is sugar additive. Acha. Now the first one. What happens to the level of the water in the beaker after the experimental period of time? Remains same. What happens to the level of the solution? Remains the same. What is the reason for the observation? Osmotic pressure prevents the movement of the solvent into the solution. Prevents.
on which part is the osmotic pressure?
The osmotic pressure is applied here on the membrane, always on the barrier.
So we will write osmotic pressure applied on the cellophane paper or that membrane.
You don't have to write applied air. The pressure which is exerted, it is understood. The pressure that is exerted is always on the other side of the membrane, other side of the barrier. What is the reason for the observation? Again, you are supposed to write. I repeat. The reason is osmotic pressure, which prevents movement of the solvent into the solution.
If the position is switched, ma'am, like in the funnel it is water and outside is sugar solution. So the pressure will facilitate osmosis.
If here you take water and here you are taking sugar solution, then the water should run out. In that case also you will come across, if it is a concept of osmotic pressure, in that case also the movement of the water molecule because of the pressure will be negligible, which smoothly should have happened. Smoothly should have happened. But if we change this membrane, then it's going to be very smooth. But we are not talking about changing the cellophane or the carton to something else. Selective permeable membrane only. With that we experiment, then the movement will be negligible that way also. Will there be any change in the taste of the water of the beaker after experimental time? Taste of the beaker, taste of the water in the beaker होने के लिए क्या जरूरी है? Taste of the water in the beaker होने के लिए this solution should flow out. No, that will not happen. Name and define the pressure. Define the pressure. Define the pressure. What did I say just now?
The minimum pressure.
Learn the definitions. Definitions हमेशा आता है पेपर में. You should know the definitions very correctly.
Minimum pressure which prevents the movement of
Solvent
solvent to the solution.
into the solution. Movement of solvent into the solution. So that is what is called osmotic pressure. Now next is when we are talking about the concept of active transport. Now let us learn another phenomena. So far we were all talking about the concept of osmosis. Now we are talking about active transport, completely different concept that we try understanding now. What is active transport? Active transport is another phenomena, and the definition of active transport, the properties of this process, the properties of this phenomena is a bit different. Here we are talking about the movement of molecules. The movement of the molecules, the movement of the molecules is from a region of low concentration, to a region of high concentration. So remember this when we are talking about active transport. Let us talk about a plant cell. This is a plant cell, the vacuole. The plant cell, surrounding the plant cell in the soil, water, there is adequate amount of ions. Surrounding in the soil, water, there are adequate amount of ions. Nitrates are there, sulfates are there. Various different type of minerals which are very much necessary for the formation of all the different type of pigments and proteins and enzymes that the plants needs. वैसे तो when I talk about the cell cytoplasm, the cell cytoplasm already has this in adequate amount. But still, the plant always wants to keep them as reserve. Because the plants always are very much in need of all these sulfates, nitrates, chlorides, various different type of mineral ions and radicals, all of these. Already the cell cytoplasm has it in excess. But still, it tries to take it from the cytoplasm because they are constantly used up. Now, in this case, I'm saying, then if that is the story, where I understand that if I'm talking about the nitrates, already nitrates are so much in the cell cytoplasm as compared to what is there in the soil. Nitrates are already there so much in the cell cytoplasm as compared to what is there in the soil. Then should this movement be an easy, simple, passive movement? No, this cannot be a simple passive movement. This cannot be a simple diffusion. So in this case, since the molecules are moving from a region of lower concentration to a region of higher concentration, this process needs ATP. This process needs the formation of ATP, or this process always requires the utilization of ATP. So this process always requires, I should write utilization of ATP, first of all the formation and then the utilization actually, First of all, the ATP formation has to take place in the mitochondria of the cells and then its utilization where the ATP is broken down. So apart from this feature, what should we also write? What is happening extra here? Number one, the molecules are not moving from a region of from a region to high to low. Since the molecules are not moving from a region of high to low, the cell has to spend extra energy molecule for grabbing them or taking them against the concentration gradient. मतलब lower concentration से ज़्यादा concentration वाले area में जा रहा है. If I say along the concentration gradient, मतलब ज़्यादा concentration से lower concentration वाले में जा रहा है, which is an easy movement always. When we go down the steps, it's an easy movement. Something like diffusion, higher to lower, it's always an easy movement. But when we have to climb the steps, it's a difficult movement. We are moving from lower to higher. So same is the concept here. When I'm talking about the cells are taking these ions and minerals and all these different type of radicals from a region of lower to a region of higher concentration and utilizing ATP. But the question is, why does the cell do this? You get questions. Give reason. Why does the cell do this? The cell is always engaged in the process of active transport also because it always keeps a reserve of the ions and radicals for the constant formation of the pigments and the proteins which are used up in its physiological processes. The chlorophyll has to be formed. So many enzymes have to be formed. The processes of photosynthesis, respiration, transportation has to go on. So this you have to understand the basic differences between diffusion, active transport. Let us write now two differences between active transport and diffusion. Make a column. Go. Active transport में ये याद रखो. Active transport में the cell is taking, the plant cell is taking all these minerals and ions from the soil, but from a region of low concentration to high concentration. मतलब you already must be having 20 t-shirts in your cupboard, इससे भी ज़्यादा. But फिर भी जब कहीं जाते हो, again you pick up 2, again you pick up 3. So you are already having surplus, but still we keep on taking. Why? क्योंकि हमें लगता है नहीं, कभी अगर जरूरी है तो एक दो नया ड्रेसेस तो होना ही चाहिए हमारे पास. So that is what the cell thinks. That is what the cell is thinking. The cell should never fall short of these ions and minerals because it is constantly necessary for the process of formation of the enzymes and the proteins and the pigments. नहीं तो उसका physiological process रुक जाएगा. So that the cell is constantly. So इसके लिए quite a number of times it is also ready to spend extra energy molecule. Like you are ready to spend any amount of money that your parents, you may brush off their pockets. नहीं मुझे चाहिए. So what is happening? The cell wants to spend all these energy molecules, but will be nitrates and sulfates and all these molecules the cell picks up. So that is the concept of active transport. But let us write down two differences between active transport and passive transport or diffusion. You get that way. Question you will get in your paper, differences between active transport or passive transport. Passive transport is the same as diffusion. Let us write two differences. Number one, the first one you write on your own. I will tell you only what to write. The first one you write down the definition. Active transport, passive transport, bracket में condition रहेगा definition, bracket में condition रहेगा energy requirement. So let us write down active transport. Write down the process of movement of molecules, process of movement of molecules or minerals and ions. Now, can we write process of movement of molecules of a substance? Basically, can we write? No, we cannot actually, because active transport is majorly not applicable for gases. Active transport is also not applicable for water. Active transport is majorly applicable for all the special type of ions and minerals. So movement of molecules of ions and minerals from a region of low concentration to a region of high concentration. From a region of low concentration to a region of high concentration. From a region of low concentration to a region of high concentration. Other side you write osmosis, you already know the definition. Huh? What is it?
In this, we will use the word water molecule.
No, I'm sorry. I said, what is it? Active transport and diffusion, no passive transport. For passive transport, whenever you should write molecules of a substance, because that is applicable for solute, liquids and gases, all. So for same thing. Diffusion and passive transport is same thing. No energy. So that's the second point that you will write. So for passive transport or diffusion, no expenditure of energy by the cell. No expenditure of energy by the cell. For active transport, extra expenditure of energy takes place by the cell. Extra expenditure of energy. When are you going to improve your writing after the exam is over? Acha, this is fast handwriting, slow handwriting, medium. How is also handwriting? कितनी तरह से handwriting आता है तुमको? हाँ, First point ये लिखोगे, the directional movement, no? Active transport, movement of water, movement of molecules of ions and minerals from a region of low concentration to a region of high concentration. Passive, passive में क्या लिखोगे? Higher concentration to low concentration. वो सबको आता है. Come to the next point. The next point for active transport you will write the cell, or there is extra expenditure of energy. Extra expenditure of energy in the form of ATP. So always, whenever such a process happens, ATP is broken down to ADP and inorganic phosphate. ATP पता है? What is the full form? I hope all of you remember that. Triphosphate. And ADP? Adenosine diphosphate. And ये phosphate free है. Anti-diuretic phosphate. You are making a hybrid of everything. So this is adenosine triphosphate, adenosine diphosphate, and this is free phosphate. Free phosphate, and adequate amount of energy is needed for the pickup. Expenditure of energy in the form of ATP, and for diffusion there is no expenditure of energy. There is no expenditure of energy. Now, next is we talk about some terms known as turgidity and flaccidity. Turgidity and flaccidity. What is a turgid condition? What is a flaccid condition? So now I talk about a plant cell, and if this plant cell has surrounding, the plant cell is now surrounded by a hypotonic medium, a hypotonic solution. The plant cell is surrounded. I have never seen him. Never ever I have seen him looking at the board and concentration से कभी सुनने की, कभी सीखने की उसमें.
पूछा था क्या बात है?
हाँ, वो ज़्यादा important है ना.
नहीं मैं सुधारा मैं क्या करूँ?
तुम क्या कर सकते हो? Of course, क्या कर सकते हो तुम? Your parents sleeps in peace at night.
एक बार बुलाया तभी देखा.
Now, hypotonic solution. Try to answer this question that I'm asking you, all of you. This is a cell, a plant cell, and surrounding the plant cell is the hypotonic solution. What type of osmosis should happen in this plant, in this cell? Endoosmosis. So yes, we are talking about the process of endoosmosis happening. अब जब endoosmosis हो रहा है, initially what will happen? The cell is constantly taking in water. The cell is constantly taking in water. So what is the first step? The first step is the vacuole is extending. What is the vacuole membrane known as? Who remembers? Vacuole का जो very good, tonoplast. ये जो tonoplast है, this is to a great extent flexible. Huh? Why like that? टोनो, ये तुमको आर दिख रहा है, मेरा इतना अच्छा हैंडराइटिंग तुम क्रिटिसाइज कर रहे हो. टोनोप्लास्ट, टोनोप्लास्ट. क्योंकि ये लोग मन से पढ़ते हैं, तुम्हारे जैसे नहीं पढ़ते. Good, very good. So now when we are talking about tonoplast, what is it? This membrane of the vacuole, it has got a higher degree of extensibility, flexibility. So इसके वजह से now when the water will start entering, is the cell right away going to get expanded or swelled up? No, it will happen stage by stage. बहुत देर में. So initially what will happen? ये जो vacuole है, this vacuole will start becoming extending, and this vacuole will now start becoming getting expanded. So say suppose I'm talking about after quite a long period of time, what has happened? The vacuole has become expanded. The vacuole is now tight filled with water. We have been keeping this cell in the hypotonic solution for quite a long time, and after this now what do we observe? That the cell has been absorbing water, absorbing water, absorbing water, And a time comes when the cell is not able to absorb any more water. No, the movement of the water more or less has stopped. The movement of the water more or less has stopped. Now, why the movement of the water more or less has stopped? Because the vacuole has reached to its last extreme of absorbing water, and now it cannot absorb any more water. It cannot absorb any more water. But again, are these cells going to burst out immediately? No, then also the cell will immediately not burst out. What is some of the real conditions that we can cite here to understand? We may come across that all these aquatic plants which are already in a hypotonic medium every time. No, the aquatic plants that I talk about. The aquatic plants are always in a hypotonic medium. Now that they are in the hypotonic medium, if I especially talk about the plants in the lake and the pond, they are of course in a hypotonic medium always. But then did we ever see that these plants which are in the aquatic and hypotonic medium, are they always bursting out or are they easily dying out? No, they are surviving for months. They are always surviving in the entire season. Say suppose you talk about the lotus. No, in the season they are always there. Then how is it possible? The water must be constantly entering their cells. Then why does these cells does not burst out and why does this plant easily doesn't get died out? No, they don't get died out because of certain factors which is constantly working. So first of all, I said after a particular period of time, initially now what's happening? Initially the process of osmosis happens, and after a particular period of time, process of osmosis slows down. Why? Because the vacuole is fully distended. So what are some of the words you are supposed to remember here? The vacuole is distended. The vacuole is distended. What is the meaning of distended? Tight filled with water. The vacuole is tight filled with water now. Then when we are talking about what is the condition of the cell now known to be? The condition of the cell is now known to be turgid. The cell is in the turgid condition. How do we define what is turgid condition of a plant cell? And how does the plant parts look like? If I have to talk about the turgid condition. In the turgid condition, the plant parts will look very fresh and it will look very fresh and stiff. Say suppose I talk about the small tulsi plants that you have at home, the money plants that you have at home. If I'm talking about that when they are well watered and if the temperature is not very high, if you're talking about the morning hours of the day or the evening hours of the day, you will see that the stem is erect, the leaves are all spread out, the leaves appear a little bit shiny also. If I talk especially about the money plants and all, the leaves appear a little bit shiny and the leaves are all spread out. The leaves are all spread out. So now in this condition, if I talk about that, if I look into the leaves all should be spread out. But the same condition do I get to see if it is afternoon, hot summer sunny day? Will it happen the same? No, on a hot summer sunny day, this is not happening because it is going to lose so much of water. This is going to lose so much of water. Then what will be the condition? The condition of the leaves would be something like this. They will become limp or wilted. They will become limp or wilted. What has happened? Initially, the vacuole was distended, was filled with water. In that condition, we had said that the cell is turgid. The cell is turgid. And the phenomena, the process, turgidity, the condition of the cell, turgid. But when this cell starts giving away water excessively, the vacuole again starts shrinking, and that may happen due to environmental condition. If the soil is not watered, either of the two may be responsible for the loss of the water from the vacuole, from the cells, and that will result in what? Wilting. And this condition of the cell would be somewhere like when I talk about the age of plant cell here. This plant cell, yes, the vacuole has started giving away so much of water, and if this is the condition, this is what is known as the flaccid condition of the cell. Then what are the differences between these two terms we are talking about? If a cell undergoes gradual endoosmosis, then the cell is gradually becoming turgid. Gradual endoosmosis will make the cell turgid. Then what should you associate with flaccidity? You have to always associate this term. Gradual exosmosis will make the cell flaccid. But here, one more thing we have to also understand, as I said, these are the two opposites. First of all, you understand what is turgid, what is flaccid. The process known as turgidity, phenomena known as turgidity, flaccidity. But now something more that you have to come into concept here is that why in this condition the cell did not burst out. Why in this condition the cell did not burst out? Because the cell was having so much of sap inside. But in this condition, there will be a particular pressure exerting from the walls of the cell towards the cell contents. Then this should actually be responsible for squeezing the cell or bursting the cell. But no, this is not actually allowing this process, because there is also a counter process acting, the process of exerting pressure from the cell contents to the cell wall. I repeat what I said. When a cell is fully turgid, are we supposed to always understand that the moment the cell is turgid, it should burst out? No, it should not. Or maybe not. Mostly not. Why not? Because at this turgid condition, two forces, counteracting forces, start acting on a cell. What are the two counteracting forces? One force or pressure which is acting from the cell wall to the center of the cell or the cell contents, which is known as Turgor pressure, So that's the first pressure. Let's define this. Turgor pressure. What is Turgor pressure? The pressure of the cell wall on the cell contents. The pressure of the cell wall on the cell contents, which is actually trying to prevent the bursting of the cell, which may have majorly happened due to this pressure. What is this pressure? Pressure of the cell contents on the cell wall. इसकी वजह से cell ज़्यादातर burst होता है. So what is this pressure I'm saying? The second pressure is known as wall pressure. What is wall pressure? The pressure of the cell contents on the cell wall. So I make it this way, that when the cell is turgid, सबसे पहले कौन सा pressure exert होता है? Pressure of the cell contents on the cell wall. Pressure of the cell contents on the cell wall, इसको बोलेंगे. Pressure of the cell contents on the cell wall, इसको बोलेंगे. Pressure of the cell contents on the cell wall, this is what is known as Turgor pressure. And pressure of the cell wall on the cell contents is known as wall pressure. So इसकी वजह से, since they are counteracting, they are not allowing the cell to burst out. And when the surrounding changes little bit, the cell again establishes its normal state. So first of all, you have to associate with all these terms. Get associated. Number one, turgidity, flaccidity क्या है? When do they occur? How will you relate turgidity and flaccidity with terms like endoosmosis and exosmosis? And what is the basic usage or benefit that a cell enjoys because of these two pressures? We'll continue again. Okay. Go through the exercises that you have in your textbook. Go through the exercises, because the next class again I will give you some set of questions that you have to solve.
अबे इसके Yes, जो 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 दिखा है ना, इरेज, मैं तो लिखा है